in terms of what we've been doing in the past. So the first point is going to be DNA and scientists. And for the DNA and scientists, let me go over to that right now so you can see. There's a PowerPoint. I'm not going to teach this. I'm not going to do a voice cast. I'm going to focus on replication. So this PowerPoint is already online, and you can just go through it. And you are responsible for knowing a timeline of events. You should be able to start from sometime in the 1800s and move up to 2020, I guess. And you should be able to tell me the series of scientists who've contributed genetic knowledge to bring us to the point that we are at now. So it starts in 1865 with Mendel, which we talk about in grade 11 biology, and then each slide here talks about a different scientist. In most cases with these scientists, it will also either superficially or with maybe a little bit more detail talk about the experiment that they did that gave us more information. So the chromosomal theory of Boveri, the Griffith experiments, the Phoebus-Levene and what he contributed, McCarty-McLeod and what he contributed, Chargaff, Hershey and Chase, Wilkins, Rosalind Franklin is a very interesting historical story. I put a really big blurb here so you can read about it. Linus Pauling, whose model was completely wrong, but a lot of times it's the wrong things that help us move in the right direction. And of course, ending with James Watson and Francis Crick, who basically, most of the history books will tell us they're the ones responsible for telling us that the DNA is a double helix and figuring out what the actual structure looks like. As you go through this PowerPoint, you'll see a number of links. And so I encourage you to click on those links. I encourage you to click on these links and try them out. And they'll bring you to places where you'll either get YouTube videos or little activities that you can kind of work through the learning process that they did as well. So that's it. That's a scientist. Look at the PowerPoint, make a timeline. If I were doing a proper test, there would have definitely have been a timeline question asking you to tell me about the series of events that led to our modern double helix. The next concept is the DNA superstructure. So DNA, evidently you can stretch it to the Earth and the Moon like 6 times, or go around the planet like 25 times, or something silly like that. There's a lot of DNA inside your body. How is it possible to get that much DNA inside your body? So there's a whole series of structures. We start from the nucleotide. The nucleotide can be bonded with a sugar to form a nucleoside. That could be bonded with a phosphate to form a nucleotide. And you take a whole bunch of nucleotides together and you get a nucleic acid. So that is all stuff from biochemistry from the first unit. We covered that already. Then we get into some serious DNA structure, and you'll notice that I have it's superficial in this note. I don't expect you to go in all the detail. If you guys looked it up and you looked at some university text or you went to some sophisticated animations, you'd see all sorts of really complex words and concepts. This, what I have on the screen here, is what you need to know. Okay, so you should understand how DNA is bound and compressed so we can fit Earth to the Moon 6 times and back and forth in our little tiny bodies compared to that distance I'm talking about. Okay, so you can use both these diagrams I have up here. They're both very similar, but the text is the same, and that is your DNA superstructure.